Welcome to Blue Oak Nutraceuticals. This presentation was first given at the 25th anniversary of the Bryan Miller Symposium at UC Davis. The following slides reference clinical studies involving mitocatalyst, the molecule and active ingredient in the mitochondrial supplement Mitocatalyst E by Blue Oak Nutraceuticals. These clinical studies were conducted on specific individuals in a limited population setting for limited periods of time. Any reference to these clinical studies is not intended to be representative of results that should be expected from the use of mitocatalyst for anyone in the general population. Children under 18 and individuals with a known medical condition should not take this product without first consulting a physician. These statements have not been evaluated by the Food and Drug Administration, and this product is not intended to diagnose, treat, cure, or prevent any disease.
Good morning, everyone. Let's talk about mitochondria and the discovery and role of a new steroid today. Next slide. To start the conversation, I want to take us all back about 2 billion years ago to an event that was proposed to have occurred called endosymbiosis. It was proposed by biologist Lynn Margulis in the 1960s, and the endosymbiotic theory states that some of the organelles in today's eukaryotic cells were once prokaryotic cells. Eukaryotic cells, probably like amoeba cell, got nutrients by phagocytosis, ingestion of other cells by the eukaryotic cells. And some of these phagocytosed prokaryotic cells survived and developed a symbiotic relationship with the eukaryotic cell. When photosynthetic prokaryotes were ingested by the eukaryotes, these prokaryotes eventually became the chloroplasts and became part of the plant kingdom that we have on planet Earth. And when the prokaryotes capable of aerobic respiration were ingested, they became what we call today the mitochondria and are an integral part of the animal kingdom, including marine life and others. And this symbiosis resulted in the host cell providing the endosymbionts a safe place to thrive, and in return, the endosymbionts worked and provided energy to the host cell. Next slide. Now, the mitochondria is a prokaryotic cell. They have their own cell membranes, just like a prokaryotic cell. They have their own DNA, and each mitochondria has its own circular DNA, like a bacteria shown in this picture, but smaller. Mitochondria, like other prokaryotes and bacterium, reproduces by pinching in half. And then eventually what happened, these prokaryotes that became part of the eukaryotes lost their capability to grow outside their host cell. So if you take a current eukaryotic cell and you remove all the mitochondria from that cell, the cells can't regenerate that mitochondria from scratch. So it is an integral part of the eukaryotic system, and all eukaryotic system cells have that. In humans, the only cell that doesn't have it is the red blood cell. So under the endosymbiosis story, one can provide an alternate explanation for evolutionary processes that are beyond the gene. And this evolutionary explanation would include metabolism and energy as a central organizing feature of life. So unlike genetic lineage, this focuses on biochemical networks rather than genes, on phenotypic interactions rather than informational inheritance. The endosymbiotic view of evolution, hence, are valuable for expanding evolutionary expansions, but it's not an alternate. Hence, it coexists with the genetic theory of evolution. After all, energy is the primal force and a basic component of the universe. Next slide. Mitochondria is fundamental to life and health, as they provide the essential energy for cells to do their function and for cells to survive. And it's the only component in your cell that can make more of itself when needed, fix itself when broken, and get rid of itself when not needed. And this process maintains the cellular hormesis, a balance, energy balance within the cell. One of the ways that you make more mitochondria is through biogenesis. Biogenesis implication is you inherently make more mitochondria through internal cellular processes. And to this day, exercise is the only known physiological stimulus of mitochondrial biogenesis that we know of. And this is something to keep in mind as I go further into this talk. Now, mitochondria have a half-life, but they can also be destroyed. And the mechanism of the destruction of mitochondria is through the permeability transition pores caused by high cellular calcium levels or glucose, or resulting from oxidative injury or ischemic events such as low blood flow you see in myocardial infarction or cerebral strokes. And the purpose of the mitochondria is to take in nutrients, food that we consume, and turn it into energy by making ATP. ATP is a compound called adenosine triphosphate and is made by the mitochondria, which fuels all cellular activities. And hence, some people call the mitochondria the powerhouse of the cell. Some interesting facts about the mitochondria. Adult humans have about 100,000 trillion mitochondria. And one single cell has anywhere between 1,000 to 2,500 mitochondria, which constitutes 25% of the cell volume. An average cell uses about 10 billion ATP per day. And ATP cannot be stored, hence it must be produced every day and every second of every day. Hence, every cell in your body, other than the red blood cell, are constantly using ATP and making ATP. And I mean that, every cell in your body. And a healthy person at rest produces their body weight in ATP every day, which would approximate to 150 to 160 pounds if this were to taken out of your body and weighed. When you exercise, and at maximal exercise, your ATP production can go up to 0.500 to a kilogram per minute because of the increased demand, energy demand of the body. The mitochondria ramp up and start producing more ATP. A typical adult hence needs about 10 to the 25 ATP per day, which they produce every day. And ATP and ADP are quintessential recyclers, so ATP is recycled from about ADP 1,000 times per day. Next slide. Another interesting fact about the mitochondria is it only comes from the mother. So if you were to determine your undisputed lineage, it would come through your mitochondrial DNA, through your maternal side, not your paternal side, which includes nuclear DNA and inherited from all ancestors, not just your father and mother, but your ancestors before that. Next slide. Now, mitochondria has been associated with aging, and people have reviewed this and published on it. And as you can see here, with age, you can see a very clear reduction of mitochondrial proteins, COX-3, COX-4, and the mitochondrial DNA content of a cell. And you also see an increased amount of damaged mitochondrial DNA. So as we age, we not only lose mitochondrial DNA, but we end up damaging, accumulating more damaged mitochondrial DNA. And this leads to a mean loss of about mitochondrial mass of 10 to 15% per decade after, say, age 21. And this leads to muscle atrophy, heart failure, loss of cognition with age, etc. Hence, aging is a state of progressive systemic loss of mitochondria, causing progressive systemic dysfunction. Next slide. This work was done and published by Singh et al., where they took normal mice and genetically turned off mitochondrial biogenesis, not completely, but significantly. And in 2 months saw this aged phenotype that you see in the middle. And when the mitochondrial biogenesis was restarted and mitochondrial levels were reestablished, you could see the younger phenotype again. Next slide. In summary, mitochondria is essential for all biological functions. It's not just the primary organs I show here, but essentially entire human physiological functions. Next slide. Since mitochondria have their own genome, and you also have the nuclear genome, the mitochondria are affected through both primary mitochondrial diseases and as a secondary effect on the mitochondria from other chronic diseases. Hence, they have pathology across 2 genomes as from primary and secondary effects. Next slide. Let's take a look at a new concept and role of mitochondria. Salugenesis, word that comes from salus, health, genesis is creation, is a process of healing and is hardwired into our metabolism and is supported by lifestyle factors such as exercise. It is evolutionarily conserved onto genetic sequences of molecular, cellular organ systems and behavioral changes that is used by living systems to heal. In essence, when we have a cut or we get an infection, your body uses its internal processes to heal itself. And for this, it needs energy and resource. And since it's genetically programmed, it's a responsive element of your body to heal itself. And the recovery, and it begins with mitochondria in the cell. Hence, the mitochondria are essential to provide the needed energy for salugenesis to take place. So, mitochondrial pathways restore health, and they're agnostic to the chronic illnesses. So, when you have an acute or a chronic illness, the mitochondria respond to that to try and heal the body. Hence, understanding mitochondrial role may identify processes to address chronic illnesses, such as helping cells to move to anti-inflammatory stage, we may see restoration of health. And with the role with this process plays in aging, as I just mentioned earlier, we may see prolongation of life as well. Next slide. So let's talk a little about chronic diseases. So current therapeutic approach means that we come against a disease. That is the practice of allopathy. Allopathy means allos, other, pathia, or suffering of disease. And it treats the symptoms with its opposite effect. So if you have an infection, we give you anti-infective. But it does not address necessarily the causality of the disease when you're talking about chronic illness. So it serves us well for acute diseases. And hence, for anti-tools, at best, they slow the decline of disease. So chronic disease is a systems problem caused by the combined action of pathogenic triggers and anthropogenic factors, and they affect mitochondrial function because it is happening over time. Energy and metabolic resources are provided by mitochondria and metabolic transformations. Hence, with this chronic insult that drive the cell danger response and create the healing cycle through excess production of ATP, ADP, or other tools to address to support the healing cycle. But when there is an abnormal persistence of any phase of the cell danger response, the system gets overwhelmed and it inhibits healing. It creates dysfunctional cellular mosaics and causes symptoms of chronic diseases, and can also accelerate processes of aging. So the approach of our healing or salutogenesis gets disrupted in instances of chronic illnesses because of the continuing chronic insult or within the cell. Next slide. So let's take another diversion here, and I will try to sum up and put all these concepts together at later stage for everybody. But let's talk about cacao, and I'm sure you'll wonder why. But if you indulge me, let's start with understanding where cacao came from. It was domesticated about 5,000 years ago in present-day Ecuador. And in 1600 BC to 400 BC, the Olmec Indians of Mexico started using cacao beans. 250 to 950 AD, the Mayans of Mexico and Central America made the drink out of cacao, and the Aztecs used cacao for trade and money. And the rest follows European introduction and industrialization. Next slide. Now, interestingly, Linnaeus in 1735 designated cacao as theobroma, or food of gods, perhaps with an insight that hopefully I will try and justify. In Mayan culture, as I mentioned, they used cacao as a drink. Two groups were allowed to ingest cacao preparation: the royal family and the messengers. They carried message from village to village. It's an intriguing situation as to why these messengers were allowed cacao when this was only consumed by royal family other than the messengers. And I will come back to that perhaps a little later in the talk. Cacao contains about 50% fat, protein about 20%, and other components such as vitamins, minerals, theobromine, and amongst them are also polyphenols. And one polyphenol in cacao is epicatechin. Now let's move to present day. Next slide. In about 2005, 2007, researchers at UC San Diego were doing a clinical trial in patients with diabetic cardiomyopathy. It was a 3-month study with 100 milligrams per day of a chocolate enriched with epicatechin. And after 3 months, they observed this in the electron microscopic evaluation of muscle. And one is very clear from the left panel to the right that post-epicatechin dosing, these sarcomeres were very well aligned and show significant presence of mitochondria. So here you have a situation where a specific phenol that is present in cacao is inducing mitochondrial biogenesis and also reforming the sarcomeres in these patients, human patients. And as I have mentioned earlier, mitochondria play a significant role in healing. And I've mentioned to you about cacao and minus epicatechin. Hence, here what we see is that minus epicatechin singularly and very potently enhances mitochondrial biogenesis. Next slide. This is now more. So looking at the biomarkers, we see increased mitochondrial proteins from biopsy samples of these patients. Next slide. And then we see increased sarcodistroglycan complexes. And this I will touch again related to applications on dystrophy and other muscle disorders. Next slide. You see modulators of muscle differentiation, such as increased expressions of follistatin, MyoD, myogenin, and an inhibition on the expression of myostatin, very relevant for muscle differentiation. Next. You see also enhanced regulators of mitochondrial biogenesis, such as PGC1 alpha, and a decrease in acetylated PGC1 alpha, supporting the EMs that we saw of enhanced mitochondrial biogenesis. Next slide. We also see a significant effect on nNOS, pNOS, and cyclic GMP levels. Next slide. And also a reduction of beta-galactosidase, a marker of senescence, perhaps indicating a arrest of progression of aging. Next slide. We also looked at, and subsequently, a single metabolic dose in fasted volunteers, one single dose of 100 milligrams of epicatechin. And what we saw was a very significant effect on the fat oxidation of these patients. And the change is shown here, where it went anywhere from 30% to a 37% increase in fatty acid oxidation in these patients. Next slide. And we did a subsequent study in middle-aged volunteers, NF1, where they were given minus epicatechin 50 milligrams twice a day for 1 week. And we saw a statistically significant increase in hand strength by dynamometer measurements, both for the right and the left hand. And also we saw again an increased follistatin expression and reduction in myostatin in these treated volunteers. Next slide. We then opened up an open-label POC study in Becker's muscular dystrophy and to study the effects of minus epicatechin both on muscle and the cardiac function. So we see here also increased muscle transcription factors, shown here. Next slide. Mitochondrial transcription factors and cristae proteins, next slide, which are part of the mitochondria. Increased sarcoglycan proteins, pretty much a replication of what we saw in the diabetic cardiomyopathy study. But here now we are treating the patients with pure minus epicatechin. Next slide. This is now markers of muscle regeneration. We see again an increased levels of follistatin, decreased levels of myostatin, increased level of MIF5. Next slide. And in an electron microscopy of the biopsy, we see before and after effects. The top, we see the cristae per mitochondria has gone up, and cristae is where ATP is made. The bottom panel, you see increased level of mitochondrial circled in yellow, and you also see that the sarcomeres that are thin have started to become thicker as an improvement for force transmission. Next slide. And what we were really surprised and very excited to see was the increase or improvement in cardiac function in these patients. And the top right panel is telling data where we see that the dotted line is post 8-week treatment, the solid line is the start. And what you can see is that increasing workload does not increase heart rate the same level as it did prior to treatment, suggesting improved cardiac output. And if cardiac output is improving and the heart rate is not, it would imply that the stroke volume or the heart function is increasing. On the left panel, you see that reduced lactic acidosis, and also the VO2 per kilogram is also significantly improved. Next slide. We also did some additional clinical studies, and we have seen no SAEs, both in healthy volunteers and patients with metabolic syndrome, which was a double-blind placebo control, which lowered triglycerides and improved their diabetic HOMA index. Next slide. Minus epicatechin coming from cacao, its pharmacology and clinical effect in multiple clinical trials. The question still remains, how does minus epicatechin induce mitochondrial biogenesis? What we found that this effect on the mitochondrial biogenesis is unique to minus epicatechin and is not seen in variety or number of its congeners and closely related polyphenols. With that data, we hypothesized that if exercise is the only way we know of how your body can induce mitochondrial biogenesis, but now we see minus epicatechin doing the same. Hence, it's mimicking some endogenous process that is perhaps induced when one exercises. Looking at structural equivalence with minus epicatechin, we hypothesized that perhaps it's a steroid with the right functional substitutions that minus epicatechin is mimicking. Doing computational studies and analysis and structural homology analysis, we proposed that 11 beta OHP, it may perhaps be the endogenous steroid hormone that induces mitochondrial biogenesis and is produced by the mitochondria or your cell when you exercise. We synthetically made 11 beta OHP and tested its effects in cells. And here we see a very clear and a potent induction of its effect on increasing electron transport chain complexes of the mitochondria in the absence of cell proliferation. As you will note, the activity is down to the femtomolar range. Hence, it's a very potent inducer of mitochondrial biogenesis. Next slide. We further looked into the effect of mitochondrial transcriptional factors, and this slide presents the effect of 11 beta OHP on various mitochondrial transcriptional factors. And as you can see, it has a potent effect also in inducing an increased expression of these transcription factors. Next slide. As I had mentioned earlier, that mitochondria are destroyed by the formation of permeability transition pores, such as that would be induced by calcium. And we looked at the effect of 11 beta OHP on induction of permeability transition pores in the presence of calcium. And this slide clearly shows that 11 beta OHP has an effect in terms of protecting the mitochondria from forming the permeability transition pores. Hence, it is the first endogenous inhibitor of MPTP that we know of. Next slide. Another marker of biogenesis is activation of AMP kinase, and 11 beta OHP also potently activates AMP kinase and at the same time inactivates ACC. Next slide. We studied at the effects of 11 beta OHP in other pharmacological outcomes, looking at intracellular fatty acid or triglyceride accumulation in HepG2 cells when treated with high levels of glucose. And as we can see, that 11 beta OHP potently inhibits the formation of triglycerides or glucose accumulation in HepG2 cells. In two in vivo models, we saw a very clear effect of 11 beta OHP when given at 0.3 milligrams per kilogram in protecting the injury, hypoxic injury, in a cardiac ischemic reperfusion model in rats. And what this data on the left shows is the area of protection or area of insult is significantly reduced when treated with 11 beta hydroxypregnenolone. And on the right is the lactate concentration is also reduced in an exercise model, suggesting enhanced mitochondrial function. Next slide. Well, to support the hypothesis, we had to show the presence of 11 beta OHP in an endogenous system. Our first assay was in isolated mitochondria, and through high resolution mass spec, we were able to show for the first time the presence of 11 beta OHP in isolated mitochondria, suggesting or providing support to our hypothesis that perhaps minus epicatechin is mimicking an endogenous steroid hormone, 11 beta OHP. Next slide. We then wanted to look for the presence of 11 beta OHP in an exercise model. As I had mentioned earlier, that exercise induces mitochondrial biogenesis. And if that be the case, does its level go up when you exercise? So this is an exercise model where rats were placed in a swimming tank, allowed to swim for 5 minutes. And on the top left panel, you see the level of 11 beta OHP increase starting at 5 minutes after exercise, peaking at 45 minutes, and within about 2 hours going back to baseline. So here was a very clear evidence of the induction of 11 beta OHP by exercises, further adding support to the hypothesis that perhaps 11 beta OHP is the endogenous factor that induces mitochondrial biogenesis. On the right, we also looked at the levels in the brain, liver, and skeletal muscle. And as you can see, that from control to post-exercise, the level in the brain, which was undetectable, goes up to 25 picograms per mil, suggesting that this hormone is present in very low levels, but since I have already mentioned to you how potent it is, perhaps that is sufficient to induce the pharmacological outcomes we see. Next slide. Over time, we have been able to show that minus epicatechin and 11-beta-hydroxypregnenolone, OHP, share a common pharmacology. This is the slide that tells you how those activities overlap, relating to then the consequential pharmacological or therapeutic value of into sarcopenia, dystrophy, neurodegeneration, and perhaps even diabetes. Next slide. The question then finally came, does it exist in humans? So far I have given you evidence of its presence in rats, induction by exercise, and also the related pharmacology to minus epicatechin. We studied, evaluated or assayed determined the OHP level of plasma in athletes at day 1 and day 2 at the same time, and saw a consistent level of beta OHP at about 1 nanomolar in these soccer players. This was also confirmed through high resolution mass spec. Next slide. We also then looked at non-athletes in a group of scientists and saw about 20 mid double-digit level, low double-digit level of the oxidized version of 11-beta OHP, perhaps a topic for discussion on another day, but it is a very related hormone, which is an oxidized version of 11-beta-hydroxypregnenolone, which is in circulation in human plasma. Next slide. Hence, here's the summary of 11-beta-OHP. It represents the new mitochondria steroid hormone. It's released by mitochondria during physiologically induced increased oxidative phosphorylation, such as exercise. And it's, as far as we know, is the first mitochondrial autocrine hormone, inducing mitochondrial biogenesis at picomolar concentrations. And it appears to exert its paracrine effects in circulating in plasma in the form of 11-beta-hydroxyprogesterone, the oxidized version, as I mentioned earlier. Its mechanism of action is through pathways that restore health via mitochondria, hence it's agnostic of the chronic illnesses. Mitochondrial dysfunction and depletion is associated with all acute and chronic diseases and could be associated with the loss of beta-hydroxypregnenolone, OHP, function, and causing a downward spiral of mitochondrial depletion and pathological consequences. Both 11-beta-OHP and minus epi share very close pharmacological properties, supporting the hypothesis based on structural relationship. There's a pleiotropic pharmacology of 11-beta-hydroxypregnenolone and minus epicatechin, and perhaps 11-beta-hydroxyprogesterone, even though they only target the mitochondria. Thus, it is reasonable to conclude that 11-beta-hydroxypregnenolone and minus epi promote salugenesis. Is 11-beta-hydroxypregnenolone the hormone of exercise? And our proposal is that based on the data we have seen and we have evaluated, that 11-beta-hydroxypregnenolone perhaps is the hormone of exercises that induces all the consequences one would see related to exercise and mitochondrial biogenesis. As to survive, adapt to stressors, particularly chronic, our bodies have evolved hormonal response systems. Hence, 11-beta-hydroxypregnenolone's role would not be unprecedented as a mediator response to non-acute stress, such as exercise and growth. Thank you.